Welcome to the 2020 annual meeting of the Society for Research on Nicotine and Tobacco. We're so thrilled to have you here on behalf of the program co-chairs, Gideon, St.Helen, Aaron McClure, Jamie Brown, and myself. We just really want to welcome you here and thank you for being here. I want to make a couple quick announcements about changes to the program. As you know, things are evolving rapidly and we're trying to monitor in real time when we have recorded sessions coming in so that we can keep as much content on the program as possible. Unfortunately, we have to cancel podium session 2, which is in this first block of sessions this morning. And there's one other change I wanted to announce, which is that we had to cancel podium session 6. But since we have some of the presenters here, we're able to move them into podium session 1. So there'll be a few presentations on flavors that will be in the session on ends, additives, salts, constituents. So we're really thrilled to have people here to present. So without further ado, I'm going to turn it over to Gideon St.Helen to introduce our plenary speaker.
Good morning, everyone. Let's try this again. Good morning, everyone. All right, that sounds much, much better. The basic science keynote plenary lecture will be delivered by Dr. Mary Heitzeg, Associate Professor in the Department of Psychiatry and Addiction Center at the University of Michigan. Now, as you're aware, the theme of this year's annual SRNT meeting is Reducing Tobacco Use in Young People, with some emphasis on understanding the underlying mechanisms of addiction to nicotine and tobacco. As we on the program committee, Andrea Valenti, Jamie Brown, who's not here, Aaron McClure, and myself, as we brainstorm on who would be the best person to give the basic science keynote, we agreed that while we wanted someone who could speak about the biology of nicotine and tobacco addiction, or tobacco dependence, that we also wanted someone who could give a broader perspective on the mechanisms of addiction across substances, so not just tobacco, not just nicotine. I'm sure you have seen that there is growing interest among SRNT attendees on co-use of tobacco and other substances such as cannabis and opioids as well as alcohol. We thought that one place to look for a person with this broad perspective of addiction is someone involved in the Adolescent Brain Cognitive Development Study, that's the ABC study. The NIH-funded ABC study is the largest long-term study of brain development and child health in the United States. We believe that we found the right person, Dr. Mary Heitzeg, to give the basic science keynote. Dr. Heitzeg's primary research focuses on developmental neuroimaging targeted at investigating two related themes relevant to adolescent substance use. The first is how early individual differences in brain function relate to susceptibility to alcohol and other drug use and problems. And 2, how substance use impacts the developing brain and, in consequence, risk trajectories across adolescents. Dr. Heitzeg is PI of the ongoing neuroimaging component of the Michigan Longitudinal study. That's a prospective high-risk study of emergence of risk and development of alcohol use disorder unrelated symptomology. She is also PI of the University of Michigan site for the ABC study, which is tracking neural and behavioral development through adolescence in over 11,000 youth across the country. Dr. Heitzeg's contribution to addiction science include understanding of 1, premorbid neural risk for substance use, 2, genetic influences on neural underpinnings of risk, 3, developmental trajectories in relation to risk for substance use disorder and consequence of substance use, and 4, neural factors underlying resilience in youth at risk for substance abuse. Dr. Heitzeg received a bachelor's degree from GMI Engineering and Management Institute in Management Systems, a master's and PhD in psychology from the University of Michigan, and postdoctoral studies in neurosciences also from the University of Michigan. Dr. Heitzeg has received numerous awards, including a 2010 NIDA Outstanding Early Career Investigator Award. We are thrilled to have Dr. Mary Heitzeg deliver the SRNT 2020 Basic Science Keynote Plenary Lecture. Welcome, Dr. Heitzeg.
Thank you for that introduction, Gideon. And I also want to thank the program committee for inviting me to speak here today. I'm really happy to have this opportunity to talk about the research that's been going on in my lab for the past several years, looking at brain mechanisms of addiction in adolescents. And I'll also be telling you about the Adolescent Brain Cognitive Development, or ABCD study, and hopefully generating some interest among all of you in analyzing some of the publicly available data. I have no disclosures. So I want to start by talking a little bit about why the adolescent period is so important when thinking about addiction. We know from national surveys that rates of drug and alcohol use rise dramatically throughout adolescence and decline thereafter. So we see these trends in these two graphs up here. So the graph on the right shows this trend for illicit drug use, and as you can see, past month use is rising steadily from age 12 and then peaks between ages 18 to 20, falling off thereafter. And then on the left, we have past month alcohol use. The red line outlines rates of binge drinking and heavy use, which again peaks in the early 20s. So these trends in substance use are really thought of as part of a wider constellation of what we think of as typical adolescent behaviors, which are generally characterized by impulsive behaviors and risk-taking. And I always use this slide to sort of show a range of these behaviors, from things that are just risky, like the guy on a skateboard, to those that are actually illegal, like drinking and driving. And I recently added this picture to capture things that are just sort of incomprehensible, the eating of Tide Pods. So why do adolescents do these impulsive, risky things that we don't really observe in children or adults? Well, over the past couple of decades, the growing accessibility of structural and functional MRI has allowed researchers to image the brains of children and adolescents in a safe and non-invasive way. So there's been much interest in understanding how brain maturation processes may lead to these adolescent typical behaviors. So a leading theory at this time points to an imbalance between two brain systems that are really critical to behavioral regulation. So these are the subcortical systems that are thought of being sort of bottom-up reactive systems that respond to emotional or rewarding stimuli, and then the top-down control system centered in the prefrontal cortex. So what is the evidence that there is an imbalance in these systems during adolescence? So let's start with the prefrontal cortex, and this is sort of the iconic figure of adolescent brain development, showing the progression of development from childhood here at age 5, which is sort of to the left, over to age 20, late adolescence, young adulthood. So in this graphic, blue and purple indicate maturity. So you can see that development is progressing from posterior to anterior, and the prefrontal cortex is the last brain region to fully mature. And even out at age 20 in this figure, you can see that it's actually not fully mature. You're still seeing a little bit of green there. So it's really maturing even further out into development. So this is an area of the brain, as I'm sure you all know, that's involved in complex decision-making, in planning, and in the control of impulses, and it's not fully developed until the mid to late 20s. So the blue line here is sort of a gross illustration of this slow, linear developmental progression of this area from childhood to adulthood. And then the second important brain system is the mesolimbic dopamine system, which is responsive to natural rewards as well as drugs of abuse. The ventral striatum, including the nucleus accumbens, is a key node in the system. So relevant to the topic of adolescent behavior, the ventral striatum responds to motivational significance of stimuli. It's involved in the integration of information from other brain regions in support of incentive responding, and it can really be thought of as sort of a gateway from perceptions and emotions to behavior. From animal models, we know that there are changes in dopamine receptor concentration in the system during puberty, and these changes are believed to have important implications for reward reactivity and sensation seeking in adolescence. And indeed, there's evidence in humans that ventral striatal reactivity is heightened to rewarding stimuli in adolescence when compared with children and adults. So my group investigated the activation of the ventral striatum to monetary reward from childhood to adulthood in a longitudinal design with 2 cohorts. So we had a young cohort that started at ages 8 to 12, and in the figure on the left, you can see their ventral striatal activation during their baseline scan to monetary reward. And then we had an older cohort who started scanning between the ages of 18 to 21. And on the right, you can see their ventral striatal activation to monetary reward at baseline. And then scanning was repeated every 1 to 2 years, and we collected 473 scans, which covered the ages of 8 to 27. So this figure shows a Cummins activation to the anticipation of reward across this entire age range. And we did find that there was a significant quadratic effect of age, which, as you can see, follows an inverted U shape, which is consistent with prior cross-sectional work. And I just want to point out that this actually held up when we looked at within subject repeated scans within each cohort. So this bar graph on the left shows this for the young cohort. Their baseline scans were during childhood between ages 8 to 12, and they showed an increase in ventral striatal activation from baseline to follow-up scans within subjects, and the follow-up scans were in mid-adolescence. And then the older cohort on the right had a decrease from baseline when they were later in adolescence, 18 to 20, to their follow-up in young adulthood. So you can see even within subjects, we're sort of seeing this inverted U-shaped design. So these observations taken together have led to this theory characterizing adolescent brain development as a tension between these sort of early emerging bottom-up striatal regions that express exaggerated reactivity to motivational stimuli in adolescence and this later maturing top-down cortical control region centered in the prefrontal cortex. So this imbalance results in sort of a hyperactive reward system with a poor capacity of an immature prefrontal control system to dampen it. So this imbalance model really provides a framework for understanding the typical adolescent behaviors that include impulsivity, risk-taking, and sensation-seeking, and also include the initiation and escalation of substance use. So all of this is about normal brain development and how normal brain development may lead to normative increases in risk in adolescence, right? But we know that many youth progress through this period without engaging in risky behaviors. And certainly not all youth who try substances actually go on to become addicted. So how do we think about risk for addiction in the context of these sort of normative brain developmental models? Well, it's very likely that there are individual-level behavioral, genetic, and environmental risk factors that can lead to variability in these developmental trajectories with implications for developmental outcomes, including substance use. So this is really the focus of the work that we've been doing in my lab for the past several years. So for the next part of my talk, I'm going to go through some of how those studies inform our understanding of how risk for substance use unfolds across adolescence in the context of this developmental model. So first is a prospective study focused on the prefrontal cognitive control system, specifically looking at inhibitory control. And we investigated whether the brain's ability to inhibit behaviors during childhood predicts early risky substance use during adolescence. We used a go, no-go task to probe inhibitory control. Many of you are probably familiar with it, but for those who aren't, they see a letter, one after another appears on the screen, and they're told to press the switch as fast as they can to every letter except the letter X. And the X appears 25% of the time. So they're building up this prepotent tendency to respond quickly, and then they have to inhibit that response to an infrequent target. So we did this in a group of 9 to 12-year-olds who were substance naive. And then we followed them over 4 years and got annual information on their substance use. And then we identified those youth who, after 4 years, had alcohol problems. So these youth didn't just have a drink of alcohol. They had reported having had a problem with alcohol, or having gotten drunk, or having binge drank, or they reported the initiation of illicit substance use, which at the time did include marijuana. So by 4-year follow-up, they had to have one of those things. Then they were in the risky user group, and then we had a group of matched controls who had not used any substances over that follow-up period. What we found was decreased prefrontal activation during inhibitory errors at baseline, so between the ages of 9 and 12, in those who then went on to have this early risky substance use behavior. So this graph shows the difference between the two groups. So those in red are those who went on to be the problem users, and those in blue are those who did not. So this was really the first study to report on brain activation at such an early age, predicting risky substance use behavior. However, others have reported similar findings in older youth. So there's a list of some of those here. The first 3 are prospective studies of adolescents out of Susan Tapert's lab. And these kids started in that study, I believe, when they were 12 to 14. And then the 4th is a longitudinal study from my group led by Jillian Hardy, who was a postdoc at the time and is now faculty at Michigan. And this was a longitudinal study comparing youth at high risk based on family history compared to low-risk youth. And the evidence really converges on this blunting of the normative prefrontal developmental trajectory as a vulnerability factor for risky substance use. And this is illustrated in this figure with the dotted blue line showing sort of that risky trajectory of the prefrontal cortex. We've also looked at the reward system in the context of risk. So one of our first studies investigated whether brain activation to rewards during adolescence mediates a relationship between genetic variation and problem alcohol use. For this study, we used a monetary incentive delay, or MID task. So in this task, participants see a cue, which is that first column on the left, displaying the different types of cues. So the cue says that they have the opportunity to win money, that's the middle cue, or they could lose money, or there's no money at stake, which is the neutral condition. Then there's a delay period, then a target appears on the screen. They have to press the switch while the target is up, and then they get feedback. If they were successful, they're told they won the money or they didn't lose money. If they're unsuccessful, they're told they did not win money or they lost money. This period between when the cue appears and when the target appears is an anticipation period. And when we look at the anticipation of winning money compared to anticipation for the neutral condition, we see very robust ventral striatal activation, and this has been shown across a large number of studies. So we wanted to see whether activation during the MID mediated a relationship between GABAR2 genotype and alcohol problems experienced over the 3 years after the brain scan. We were investigating GABAR2 because it's been associated with adult substance use disorder as well as adolescent externalizing behaviors, which is a robust risk factor for substance use disorder, in a number of prior studies. So what we found was that GABAR2 genotype did impact ventral striatal activation. So this effect is illustrated in this bar graph on the left. The G allele is the putative risk allele from prior association studies, and you can see here that the G allele was associated with heightened activation. So we have sort of steadily increasing ventral striatal activation in this graph based on the number of G alleles. We also found that ventral striatal activation was associated with alcohol problems over the following 3 years, and this was controlling for lifetime alcohol use up to the scan. And finally, we found that there was a significant indirect effect of GABA2 genotype on later alcohol problems via ventral striatal activation. So this suggests that risk conferred from this genetic variation may act in part through heightened activation of the reward system. More recently, we looked at prospective relationships between reward activation and substance use initiation. And this was led by Laura Cope, who was a postdoc in the lab at the time and is now faculty at Michigan. And similar to our study of inhibitory control, she asked whether brain activation to reward during childhood predicts substance use initiation in adolescence. Again, we used the MID task to look at ventral striatal activation to rewards. The youth in this study were 10 years old on average and substance naive. And then they were followed until they were 16 years old. And those who initiated any substance use were identified and then compared with a matched control group who did not initiate substance use. And what she found was that heightened ventral striatal activation to the MID task in childhood predicted those who initiated substance use prior to age 16. So we have a couple studies suggesting that heightened reward activation may be a risk factor for problem substance use. Other groups have also investigated the role of reward circuitry and risk for problem substance use. And these are primarily cross-sectional studies comparing youth with a positive family history compared to youth without a family history of substance use disorder. So one other study has found heightened activation in youth at risk based on family history. However, two others found no differences. The second one, the Muller one, is actually in a very large sample from the Imogen study, and they found no differences between family history positive and family history negative youth in a large group of 14-year-olds. Other groups have conducted cross-sectional investigations looking at the balance between reward activation and prefrontal cognitive control as a potential risk factor in family history positive youth. And these studies have been finding that those with a heightened reward activity and less control are those who have a positive family history and at greater risk. So there's evidence to suggest that heightened reactivity of the ventral striatum to reward is a risk factor for problem substance use. And this is, again, illustrated here with a dotted line, so the dotted red line. And then this is not really inconsistent with the possibility that it's actually a greater imbalance between these two systems that confers risk. So we have some idea about vulnerability at an early age and how that maps onto this developmental model. And this is really vulnerability for early initiation of substance use that we're thinking about right now. So what happens then with escalation of use when the developing brain is being exposed to substances? We've been investigating this in the context of marijuana use in our lab, so I'll be focusing on that. So again, I'll start with the prefrontal control system. We have investigated whether marijuana use impacts personality development through effects on brain functioning. So for this study, we tracked marijuana use frequency prospectively in youth from childhood out to age 20. And then based on these data, we made two groups. There was a marijuana user group made up of youth who smoked marijuana at least 100 times prior to the age of 20. And then there was a non-user group who had smoked either not at all or only a few times by age 20. And for the marijuana group, the average age of initiation of use was age 13. We also did fMRI with an emotion regulation task at age 20. And then the personality measure that we looked at is actually based on the temperament literature from Block and Block. It's resiliency, which requires some explanation. It's not the same as resilience. So resiliency is the ability to flexibly adapt one's level of control in either direction in response to the demands of the environment. So it really involves sort of thoughtful, deliberate control of behavior in challenging or stressful situations. So, for example, when taking an exam. And freer expression in circumstances where it's appropriate, such as at a party. It's also been proposed that resiliency relies on prefrontal control systems, which is why we were particularly interested in this construct. So we had resiliency measures at age 13, 20, and 23 for this study. And what we found was that the marijuana-using group had an altered trajectory of resiliency development. And you can see this in the graph on the right. So what we see is that those who were not using marijuana had this sort of steady increase in their resiliency from age 13 to age 23, and this is really what we expect. It sort of mirrors the progressive development of the prefrontal cortex. And the marijuana-using group did not show this progressive increase in resiliency. And it's also really important to point out that if we look at age 13, there is no difference between these two groups. So this suggests that it's not a difference in resiliency that was a risk factor for marijuana use. Okay, we also found that the association between marijuana group and resiliency at age 23 was mediated by prefrontal activation at age 20. So the marijuana group had less prefrontal activation in this emotion regulation task, and then less prefrontal activation was associated with less resiliency at age 23, and this mediation model was controlling also for earlier resiliency. So this study suggests that marijuana use may further blunt this altered trajectory of prefrontal development, which is shown here with the extended dotted blue line. And this is really consistent with what other people have shown with other substances, such as alcohol. And we also looked at the impact of marijuana use on the reward system. So here we're asking whether marijuana use during late adolescence and emerging adulthood impacts ventral striatal response to monetary reward. And the study was led by Megan Martz, who was a postdoc at the time and is now faculty at Michigan. Megan conducted a cross-lagged analysis using repeated measures of ventral striatal activation during the MID, marked in this graph as NACC for nucleus accumbens, and also looked at marijuana use longitudinally. So the time point she was interested in was when they were on average age 20, then 2 years later on average age 22, and 2 years later when they were on average 24. In the modeling, she used the demographic covariates of sex, baseline age, and parent history of substance use disorder. And then she also included substance use covariates, so prior marijuana use up to baseline, and prior and concurrent binge drinking and smoking. So this is what she found. These are the results of the cross-lagged model. So you see the numbers on top. You see the expected autostability of nucleus accumbens activation. So age 20 activation is correlated with age 22 activation, is correlated with age 24 activation. We see something similar with marijuana use along the bottom, so we're seeing autostability there. The interesting finding here is the cross-lagged part. So we see that if you look at the lines from nucleus accumbens to later marijuana use, we're not getting significant effects. However, when we look at the lines from marijuana use to later nucleus accumbens use, we're getting significant negative effects. So this is telling us that marijuana use is associated with decreases in later ventral striatal activation to reward over time when controlling for all prior use. So this study suggests that marijuana use may also blunt the trajectory of reward system development. And these effects of marijuana on both the cognitive control and reward systems have also been noted in a recent meta-analysis that came out a couple years ago that was led by Matt Sutherland at FIU. So looking back at this overall model, so I said more activation in the reward system is associated with risk. And then I said that marijuana use is lowering the activation in the reward system or blunting the activation in the reward system. So is this a good thing? Is this somehow adaptive? Are we happy about this? Let's take a closer look at the system. So we see the normal development in the solid red line of the reward system, this inverted U-shaped development. And then we see this risky development with a dotted red line, which is high at the initiation of use, but then we believe decreases with further use. So the important thing to be thinking about here is that what we're looking at is this system's response to money specifically. So money is pretty universally rewarding, but it's not a drug reward. So what we believe is happening is this. The purple line here represents a theoretical reward system response to drugs or to drug cues. It's basically zero prior to drug use, of course. But once we get out where the reward system, during escalation, the reward system is starting to respond more and more to drugs and drug cues, and eventually is responding more to those types of cues than natural rewards as response to natural rewards decrease. What we get is a situation where we have addiction. And this model is consistent with the idea of drugs hijacking the reward system. So it's a state where an individual has lost pleasure in those things that they used to enjoy and are motivated primarily by seeking out the drug that they use. So this has not been tested in a longitudinal design across adolescence and into adulthood in humans. However, this is a theoretical model based on incentive salience that would explain why we're seeing with continued marijuana use this blunting in a natural or universal reward, such as money. So when we put that all together, what we see is a greater imbalance between these systems that may be a risk factor for experimentation with drugs and even escalation of use. But then substance use itself further disrupts this balance. And then out under addiction, we're seeing that prefrontal control is quite low, and so is the response to natural rewards. So we think that this reward system is now responding to drug cues rather than what we would consider a more natural reward. And at the same time, the prefrontal system is compromised, which makes it difficult to control the impulse to seek out more drug rewards. So it's kind of a complex picture, but as complex as it is, it's actually a quite simplified account because there are really limitations to what we can do with the small sample sizes that we can test with a typical grant. So for example, interaction with psychopathology is not accounted for in these models. So we know that adolescence is a prime period for the onset of not only substance use, but other mental illness. So this figure is from a review by Paus and colleagues, which shows age ranges for the emergence of a number of disorders. And you can see impulse control disorders are that top bar, which emerge quite early, starting at age 5. Underneath that is substance use disorders, emerging starting around age 13. Below that, anxiety disorders, emerging overlapping with both impulse control and substance use. Below that is mood disorders, also overlapping and emerging around age 14, and then below that is schizophrenia. And there are also high rates of comorbidity between substance use and psychopathology. So this graph shows that youth between the ages of 12 and 17 who had a major depressive episode in the past year were more than twice as likely to use illicit drugs as those who did not experience major depression. So in this graph, the red bars are the total of 12 to 17-year-olds using in the past year, and then the tan bar is those with major depression, and the blue bar is those without. Another limitation is that substance-specific effects are not ideally modeled. So we know that adolescents are using multiple substances. We just know this. So in the studies that I talked about, when I said we're looking at marijuana use, what are we really doing? So so in the first study, we were trying to match the two groups on their alcohol use, because we know that if they're using marijuana, they're probably using more alcohol. How do we deal with that? So we tried to match the two groups on alcohol use. In the second study on marijuana use, as you saw, we used covariates for other substance use. But this is not an ideal way to get at substance-specific effects. Another limitation is that environmental and contextual factors are not accounted for in these models. So things like neighborhood environment, school environment, home environment, et cetera. There's just not the sample sizes to include all of those variables in these models. And finally, the samples are not representative. And there's a real lack of racial and ethnic diversity in the neuroimaging studies that exist out there right now. So ideally, to really fully understand these complex relationships, one would need to recruit a very large and diverse sample of youth prior to the initiation of substance use and follow them through adolescence. So I think you know where I'm going with this. Yes, the Adolescent Brain Cognitive Development Study is doing just this. So it's an NIH initiative to recruit over 11,800 youth when they're 9 and 10 years old, follow them over a 10-year period with extensive assessments, including but not limited to neuroimaging, genotype, and a variety of psychometrics. So this study was initiated by the NIH Collaborative Research on Addiction, or CRAN, which is comprised of NIDA, NIAAA, and the National Cancer Institute. And the objectives were really centered around these questions regarding substance use and its interactions with brain development and mental health throughout adolescence. However, the study assessments are so broad that many other federal collaborators are now participating, and this is a list of the current federal collaborators. So there are 6 broad objectives for the ABCD study: to develop national standards for normal brain development in youth, identify developmental trajectories of brain, cognitive, and emotional development, academic progress, and all of the risk and protective factors that can impact them, examine the roles of genetic versus environmental factors and their interactions on development, and this is facilitated by a large twin sample, study the effects of health, physical activity, screen time, sleep, sports injuries, other injuries on brain development and other outcomes. Now we're getting into the actual substance use goals. Study the onset and progression of mental disorders, factors that influence course or severity, and the relationship between mental disorders and substance use. And determine how exposure to various levels and patterns of alcohol, nicotine, cannabis, caffeine, basically all substances, affect developmental outcomes and vice versa. So we're trying to do a lot. So in order to do this, 21 sites across the U.S., and those sites are identified by the red pins on the map, have recruited 11,878 youth ages 9 to 10 years old, including a large twin sample and some triplets, and are following them over time. And the goal was to put together a study of youth that reflects U.S. demographics in terms of race, ethnicity, sex, and socioeconomic status. So how did we do with that? So this shows the racial and ethnic makeup of the ABCD study and compared to the American Community Survey of U.S. Households, or the ACS. So on top is the general ABCD population, and on the bottom is the twin population, and there were slightly different recruitment methods for those two populations, so we kind of talk about them separately. And the orange boxes are absolute numbers for each category, and then in the circles, it shows how well we did regarding hitting our targets, which were based on the ACS. So the first number in parentheses under each category is the ABCD percent, and then the second number is the ACS percent. So for example, for Hispanic, in the general population, we were targeting 23%, and we ended up with 22%. In the center, the percentage in the center of those circles is our overall success at matching the US population of 9 and 10-year-olds. So in our general population is a 97% match, and our twin population is a 96% match. And this shows some socioeconomic status indicators compared to the ACS. So we see household education on the left and household income on the right. And then within each of these, the bar on the left is the ABCD general population, the bar in the middle is ABC twins, and the bar on the right is ACS. So if you look over at household income, you can see that we did a pretty good job in the general population of matching the US population. For education, on the other hand, you can see that we are skewed sort of toward a higher education group, although we do have an impressive range for a neuroimaging study. Overall, we have succeeded in recruiting a large, diverse sample, and this will allow us to have a comprehensive survey of neurodevelopment. It will also allow the study of specific subgroups and matched controls. So if we think about wanting to understand substance-specific effects, for example, a small percentage of our 11,000 youth may end up using only marijuana, but a small percentage of 11,000 is probably larger than a current neuroimaging study trying to investigate substance use effects. It also allows for us to increase the rigor of our analyses. For example, we can have built-in replication, do split half samples, et cetera. And it allows for the potential to disentangle demographics that are often confounded, such as race, SES, and urbanicity. So what are we doing with these kids? We have comprehensive assessments at baseline, and then every other year, these comprehensive assessments include multimodal neuroimaging. Then in the off years, we have briefer assessments without neuroimaging. So we see these kids in person every year. And then in between those annual assessments, we have, right now, it's phone assessments, phone or web assessments at 6-month intervals. And these are really designed to capture any initiation of substance use while it may still be fresh in the youth's mind. And we're going to go to more frequent assessments for that. So these are kind of the key constructs of interest in the ABCD study. So the changes in brain, mental health, and substance use in adolescence that we've kind of already talked about. And we certainly expect that all of these changes have reciprocal effects on one another, with implications for developmental outcomes. And there are also other interacting risk and protective factors that are of interest in the ABCD study. So for example, the home environment, the family factors, and this includes not only genetics and family history, but also parenting and family relationship quality, school environment and peer relationships, things like screen time and sports and other activities, sleep quality, and even gender identity are being covered in ABCD. So this is an overview of the baseline protocol for the youth. This takes about 7 to 8 hours, and I'm not going to go into it in detail. In a little bit, I'll tell you where you can go to get all of the details. But we do get information about physical health, mental health. We do an extensive neurocognitive battery. We have a substance use assessment that I'll talk more about, and we get information about culture and environment. And of course, we do multimodal imaging with high-resolution structural scans. We get diffusion-weighted imaging to look at the integrity of white matter. We also collect up to 20 minutes of resting state data with the hopes of getting at least 12 good minutes of resting state data. And we do 3 tasks. And these tasks are really informed by these kinds of neurodevelopmental models that I've been telling you about. So we do a monetary incentive delay task, which captures this bottom-up reward system. We do a stop signal task, which, similar to the go-no-go task, captures this top-down cognitive control. And then we have a twofer in our emotional end-back task, where we're getting both cognitive control with a working memory portion, and then we also have an emotion element, so we can tease that out and just look at emotional reactivity. And we also do an assessment with the accompanying parent, which takes about 3 hours, on the youth's physical health, on mental health, substance use, culture, and environment. So in order to maximize the impact of these data, ABCD has embraced an open science model. So the data are publicly available on the NIMH Data Archive, or NDA, and they're available to all qualified researchers with the completion of a data use agreement. This is the URL. We have 2 types of data available. Fast track data is unprocessed neuroimaging data and basic demographics. It is released on an ongoing basis. And then we have an annual curated data release, which includes all of the domains that I talked about. Release 2 happened in March of 2019 with the full baseline cohort and about half of the 1-year follow-up. And for those of you interested in these data, this gives a general idea when longitudinal data becomes available. So for example, if you look out to 2024, what you see is that full data from 6 time points will be available out to age 14 to 15. And those ages that are appearing in yellow are when the neuroimaging occurs. So in 2024, these data will include 3 waves of neuroimaging. ABCD has also developed a data exploration and analysis portal that anyone with NDA access to ABCD data can use. So this enables cloud-based interactive analysis of the data with factors pre-coded to facilitate the nested multi-level modeling that you'll need to do with ABCD data. So you really need to account for site that the data was collected at, as well as family, because along with a large sample of twins and some triplets, we also have siblings that are enrolled in the study. So for more information about the baseline protocol, a special issue of Developmental Cognitive Neuroscience came out in August of 2018, which gives detailed information on each of the assessment domains. And more recently, a paper came out in Neuroimage, giving details about image processing and analysis methods. And you can also go to the abcdstudy.org webpage, which has not only links to the publications that I just talked about, but also links to the NDA data access, as well as just more information about the study. So I just want to finish up by telling you a little more detail about our substance use assessment. So we assessed 3 main domains, and I'm going to go through each of these separately. So, whoops, I'm going to go the other way. All right. So we assess detailed substance use patterns. So we get a lot of information about low-level use, so that's sipping of alcohol, puffing of tobacco, puffing of marijuana. Then if a full use occurs, we do a timeline follow back. We get lifetime patterns of use, and then we get detailed patterns of past 6-month use. And we also get information about caffeine use. So what does use look like at baseline? So this is low-level use. So 23% said that they'd had a sip of alcohol. If they have, they're asked whether they've had a sip outside of a religious ceremony, and 17% have. In terms of tobacco, only 0.7% had had a puff of tobacco, and only 0.2% had said they had a puff of marijuana. Looking at full use for alcohol, 0.18% had had a full drink of alcohol. That translates into 21 youth from the baseline. 0.08% cigarette, 0.09% an e-cigarette or vape, 0.05% marijuana. So drug exposure is clearly minimal in this cohort, which is very consistent with the goal to characterize the youth prior to the initiation of significant substance use. So the next thing I'm going to show you is not in the data release, and I would appreciate it if it were not shared. I just kind of wanted to show you what the trends are looking like. So this is an area of particular interest, the e-cigarettes or vaping use. So this shows at baseline, 11 of the youth had endorsed this. And then it shows, and this is obviously partial data, right? So if you look at 3-year, we've only got 759 youth in that sample so far. But you can see clear escalation of these youth endorsing this kind of use. And I just want to point out, because you guys probably aren't doing this in your head, right? At baseline, they're 9 and 10 years old. At 3 year, that means they're about 12 to 13 years old, so they're in about 7th grade. And so I just want to bring that up, because the next thing I'm going to do is I'm going to pull up the Monitoring the Future numbers, which were recently released. So this is the 2019 numbers for Monitoring the Future, and the green is showing the 8th graders and nicotine vaping, which is past month use. So this is a different question than they're actually being asked in the ABCD study. So the numbers in the ABCD study, when you're getting out to 3 year, was like past year use. But you can see that these rates are steadily increasing in 8th graders, and for the 12th graders, the 2017 to 2018 jump in nicotine vaping was the largest 1 year jump that's ever been tracked in the Monitoring the Future study. So now we're looking at marijuana use and vaping, and again, this is preliminary data, looking at it over time. Very, very little, and I put it on the same scale as the nicotine, just so that you could sort of get a comparison visually. So it's increasing, but it's not increasing to the levels that the nicotine vaping is at. And again, so let's see. The vaping, so the blue bars, I should explain, are any marijuana use, and then the red bars are the vaping of marijuana. So vaping is at 0.5% for these youth, which is a lot lower than what we're seeing in the Monitoring the Future for the 8th grade. And again, I'll point out that this 2018 to 2019 increase is the second largest increase that's ever been seen in the history of the Monitoring the Future study. Okay, moving on to the second domain, that's factors that impact risk for substance use. So we get information about peer substance use, intention to use. Those are youth reports. Then the parents report on availability of substances and parent rules about substance use. I'm not going to talk about the parent report results, but again, I'm going to show you preliminary longitudinal data on the other two. So first, looking at peer substance use, how many of your friends do the following? And this just shows you the percent who say none of my friends do the following. And as you can see at baseline, those percentages, well, across the whole time frame we've collected, the percentages that say none of my friends are doing these things are quite high. But I wanted to point out what's happening with marijuana and e-cigarettes. Those two are dropping more dramatically than the others over time in terms of saying none of my friends have done this. So marijuana use has dropped from about 99% and down to about 93%. And e-cigs has dropped from about 99% to 86.5%, which is a considerable drop. And then intention to use, this is just part of the questionnaire. It comes from the PATH study. Have you ever been curious about, and this is percent somewhat to very curious, so the numbers are low, indicating they're curious about using this substance, and this shows it over time. So for drinking alcohol, it went from 1.8% at baseline to 4.1% at 3 year follow-up. For marijuana, it went from 0.7% at baseline to 3.5% at 3 year follow-up. And for tobacco products, there's a little asterisk there, which means always check the data dictionary. So the use of tobacco products has been changing pretty quickly. And so in the ABCD study, we're sort of struggling to keep up. And of course, it's always difficult with a longitudinal study, knowing when to actually change the questions. So we did start to change how we're phrasing the tobacco questions. We have collected this data. This asterisk does not mean the data does not exist. It means the data is coded under a different variable, and so I was not able to pull it quickly. But it also means that as users of ABCD data, if anyone's going to use ABCD data, it is very complicated. There are data dictionaries available, and you really need to be familiar with them. Okay, just a note that we added at 1 year follow-up, the Monitoring the Future Perceived Harm questionnaire, as well as the approval and disapproval of use from the Monitoring the Future. And we also started getting youth ratings of availability of substances. And I'm just going to show you the perceived harm real quick. For alcohol, the blue line is trying 1 or 2 drinks of an alcoholic beverage, and this is, again, over time, starting at 1 year to 3 year. Preliminary, not to be shared. But as you would expect, the percent who think there's no risk or slight risk is increasing with age, so they're considering it to be less risky as they get older for small amounts of use. But if you look at having 5 or more alcoholic drinks once or twice each weekend, so this heavier use, their perceptions of that aren't really changing, and that's the green bar. This is for marijuana, so again, it goes from low-level use in blue to regular use in green, and their perceptions that there's no risk or slight risk are actually increasing across all 3 of those categories for marijuana. And then finally, nicotine. It's cigarettes, sort of low-level to high-level risk, blue to green, and then the orange bar is using electronic nicotine or vaping products regularly. And interestingly, the vaping is actually holding steady across the ages. And finally, just a quick, this is low-level use for the 3 different types of substances, just so you can get them all on the same screen and see kind of how they're thinking about them differently. So alcohol is the least risk. Surprisingly, cigarettes is the next least, and then marijuana is like the riskiest, if you want to put it that way. All right. Okay, we also get information about consequences of substance use. This is only triggered if they use 2 or more substances in the past 6 months. They get these extra questions. I'll just say this wasn't triggered at baseline, which is good. It's consistent with the minimal use that we see at baseline. So, in conclusion, with these ABCD data, we expect that the scientific community as a whole, those interested, will be able to examine the risk and protective factors influencing trajectories of substance use and its consequences, examine the impact of detailed patterns of substance use on brain development, cognitive development, health and psychosocial outcomes, understand the interacting and reciprocal relationships among brain development, mental health, and substance use over time. And this is just sort of an example. We like cross-legged models in my lab of how you would look at those relationships over time. And then it also sort of highlights, well, it doesn't highlight, but in the bottom left, you see that with a sample this size, we can, of course, also look at sex differences in these relationships as they unfold over time, and we can also look at contextual influences. We can look at neighborhood factors, we can look at family factors, we can look at a number of factors that are influencing these relationships. And more broadly, we hope to be able to understand the biological and environmental building blocks that best contribute to successful, resilient young adults, and believe that open science will accelerate the progress and increase the impact of this work. And finally, I just want to acknowledge all of the talented postdocs and faculty collaborators who've worked on these data, as well as all of the great study coordinators and research assistants. And I also want to acknowledge the ABCD funding, as well as the ABCD data release that the data I was actually allowed to talk about was taken from. Thank you.
Excellent, very good. So we have time for a few questions.
Yeah, and then I have to catch a flight.
And she has to catch a flight. So we'll just take a couple of questions.
Yes, thanks for that really excellent presentation. And I just had a question about the overall underpinnings of the ABCD study, in the sense that it's meant to represent or try to capture representative drug use, and yet monitoring, is there a concern that monitoring the use will influence use, and how is that handled? And then the related question that, you know, if a kid says, yes, I've been shooting up heroin and smoking crack cocaine, or are they just kind of left to, okay, duly noted, and and not offered intervention. So how does that work with the ethical considerations, but without the disruption of the naturalistic data?
Sure. Okay, so first off, does monitoring use change use? One thing I'll say about that is that considering the breadth of the study, it is certainly not a focus on substance use. When I talk about it, it's a focus on substance use for sure. But for the kids participating, and if you think about the vast amount of data we're collecting, it is not a study about monitoring substance use. So even when we do those interim phone calls that I was talking about, I mean, the reason we do them is because we want to know if they're initiating substance use, but we have a whole range of other questions that we're asking, and it's sort of just folded into it. It's possible that just asking the questions about substance use may impact it, but our questions are really not about the naturalistic progression of substance use per se, but about how it interacts with mental health and brain development. And so there's no reason to believe that these kids are just not going to use substances, which then gets to your second point about what do we do when we've got kids who have serious substance use problems. And this is something, or are using serious substance use. And this is something that we have really struggled with. So certainly, as you can imagine, in our consent forms, we tell everybody that this is private, confidential information unless we think that there is a risk to you or others. And so to what extent does that include substance use? What are our ethical responsibilities regarding that? There is no plan there's no plan to offer interventions that we always give for every kid who comes through, every parent, every family who comes through. They are given a list of mental health resources in the community, and which includes substance use resources and other mental health resources. So they are all provided with those resources. There is no plan to offer interventions if we see problematic use. There is no plan to break confidentiality if we see problematic use unless we have reason to think that they are actually, there's an imminent threat. And then how do we define an imminent threat? That's tricky because you can combine various substances. You don't necessarily know what imminent threat is in this population. And so our plan is to really, we haven't gotten there yet because they're 12 and 13, is to be pretty conservative about it. If as a site, you feel like this could be risky to the kid, then we'll have to start to talk about breaking confidentiality and getting the on-call clinician involved.
Shall I go first?
Sure.
Thank you. Deborah Arnott from Action on Smoking and Health in the UK. First of all, just an awe-inspiring resource and really impressive presentation. Thank you so much. I had a couple of questions. Firstly, you are looking at twins and siblings. Are you looking at adoption as well? Is that because clearly both siblings and twins have a genetic link?
Sure, no, we're not, it's not a focus. Adoption was not exclusionary, so we do have some youth who are adopted, but it's not a focus of the study, so there was no effort to
and it's not a large enough sample to use to look at that?
Frankly, hey, get data access and look into it. Seriously, it's possible that there is a large enough sample, it's just not on my radar.
And the second question is about type of nicotine product, because perceptions of risk, if they're all folded together, it's just really difficult.
Yes.
Is it possible to separate out in particular e-cigarettes, vaping nicotine, compared to combustible tobacco?
Yes, and we are working on that.
Great.
It's in flux as
Thank you.
the importance of these things is becoming more and more clear.
Thank you very much.
Yep.
Thanks. Andrea King, University of Chicago. Hi, great talk. The first half of your talk was pretty compelling in terms of the ventral striatum and over time in development of addiction, how monetary and other rewards, the response would go down, but to drug or drug cues, it would go higher. So my obvious question is, the study doesn't seem to have drug cues in the imaging portion. And
Now it does.
Oh, it does. Can you update us on that? When are they going to be included, and what drug cues?
We just started about a year ago. Okay. And so this is a cohort that's been followed for a while. And so within all those same subjects that I was showing data on, we've now added a drug cue reactivity.
Oh.
Because of, yeah.
Yeah. Now we have the alcohol, marijuana, and tobacco are probably the top three. Are you doing all of them?
We are doing alcohol because, the whole context of that study, which I didn't mention, was the Michigan Longitudinal study, which is children of alcoholics. And of course, they're involved in a lot of substances, but alcohol is going to be number one. So there's only so much you can do. Well,
great. So you won't have them at age 9 or 10, but 11 or so?
Yeah, it'll start when they're about age 15. So that's kind of a nice time to be thinking about brain response changing to alcohol cues, because you're not going to get much with alcohol cue paradigm before they start using alcohol.
Yeah.
I mean,
a nice baseline, but yeah.
Right, but yeah, it's kind of a weird thing to, anyway.
All right.
We'll be able to see how it changes through later adolescence and early adulthood.
All right, thank you.
Linda Ferry from Loma Linda University in Southern California. I have a question about, as a clinician, I have a question about what's the community clinical standard measurement for assessing the kind of cues that you get from neuroimaging. So you're following a family, the parents are alcoholics, the parents are using marijuana, you're wanting to look at risk in these kids. Do you have an instrument or tool that is a community screening tool for pediatricians and family practice doctors to use that could give some indication the way you have shown this progression and risk? That nothing like that has been developed at this point. And hopefully through the ABCD study, we'll be able to see sort of the utility in a very large sample, right? Because I'm talking about relatively small, yes,
homogeneous samples that are indicating that this may be what's going on, but it may be what's going on in white kids in southeastern Michigan, right? So with the ABCD study should help us see how good these types of tools would be at prediction.
Mm-hmm. And the age, from your knowledge of this science in this field, the age at which you can predict is what? Do you need to wait till 13? When, from a...
how old is if you're looking at a particular youth,
yeah,
how old do they have to be for you to be able to predict what's going to happen?
Do you need to screen them at 10? Where's the age where screening is reasonable from a clinical point of view?
Right. And again, I'll say that I am not sure. I would say based on the neuroimaging data between, around 10. But, there's older studies that have shown that the behavior of 3-year-olds is predictive of later substance use, yeah. So, it could be that early. Yes.
Okay, so we'll take one more question.
Thanks. Really, really nice talk. And one thing that we're interested in is the chicken or egg question of substance use in schizophrenia. And at a 1% background rate, that's 118 kids, right? So are there any strategies that ABCD is thinking of to try to maximize the impact of that small number? Because that's like a usual imaging study then.
Yeah, right. So one thing I'll mention is that we are getting prodromal psychosis ratings at 9 and 10 years old. I think Deanna Birch she's definitely presented on it. She may have published on it. How do you maximize? I mean, that's the incidence in the population. One thing you'll be able to do is get their prodromal ratings at 9 and 10, which nobody's ever been able to do, and see how, those can be used as predictive factors and what their brains look like at that age. But even a sample size that large of 100 or so is going to be larger than any other sample size that has tracked brain development in somebody who ended up with schizophrenia.
Thank you.
Thank you very much. Okay, let's put our hands together for Dr. Heitzeg. We'll move on to the next. There's a symposium in here, and then we'll move on to the next sessions.